Good afternoon to all present here. So today our class would be on genetics. So let's see how do we come to the role of a cell to gene. So basically, tell me what is a cell? We all know cell is a basic unit of life. When we come to the structure of cell, we find that the most important part is the nucleus. What is the most important part? It's the nucleus. So when we study the nucleus in detail, we understand the nucleus is the brain of the cell. Why is it brain of the cell? Because it is performing the most of the functions of a cell, and basically we are going to know about the reproduction aspects of the nucleus. So we know each nucleus consists of a nucleosome, and in the nucleosome we are having a thread-like structure called chromatin, and a chromatin is made up of nothing but DNA and histone proteins. So this is what we know from the previous classes. Now let's see how each of them are interlinked to each other. So first comes the basic unit of life, that is the cell. Inside the cell we are having a nucleus. The nucleus is composed of chromosomes. The chromosomes are nothing but it's made up of a DNA. What is a DNA? DNA are deoxyribonucleic acid. So these DNA contain specific nucleotides. What are nucleotides? What are nucleotides? Are sequences of gene, and they are made up of four nitrogenous bases, that is adenine, thymine, cytosine, and guanine. Yes, we already know this. Now, coming to each segment of DNA, it's made up of gene. So, what is a gene? Now, gene is a sequence of nucleotides in a particular order. So, whatever our DNA or the double helix is composed of is made up of sequences of nucleotides. That means the adenine, thymine, cytosine, and guanine, they are arranged in a particular fashion. They are arranged in a particular fashion, and they are quite like giving us a particular trait, a particular characteristics, and these characteristics are known to pass from generation after generation. That is why when you see when a baby is born, right, you like if everyone starts guessing that the eyes they look like the father, or some they say no, don't, they are looking like a mother, or the hair color is like the grandfather, and the the nose looks like the grandmother. So this keeps on. It's a common discussion point when when a newborn is in front of you. So let's see how the roles of a cell to a gene and how these characteristics are passed from one offspring to the other, and it's continued from generations to generations. So you must have heard about the word heredity. So what what is actually heredity? Heredity is nothing but these gene contains each segment of a gene consists of a particular trait, and in a particular way a particular nucleotide sequence which helps us giving a characteristics or something called trait in a biological manner to the offspring. So how can we define a gene? A gene is a sequence of nucleotides in a particular order. So the in the strands like the A, T, G, C, or A, T, G, like that, it will be in a particular sequence. Let's say, for example, we have suppose in we want to see in a chromosome number one. We all know humans have 46 chromosomes in our human body. So just let say I want to study gene number one. So gene number one is having suppose 10 nucleotides. So in a human being, the first gene will be having the same 10 nucleotides for every single human being. So each DNA is a section of DNA that codes for a particular function. So it is coding for a particular function that is, it is like giving the cat. This part is giving the eye color, or this part is giving the hair color. It's like that. Okay, so they are nothing but units of hydrogen. Now, the branch of biology that helps us study about the genes, that every aspect, every particular nucleotide or every particular nitrogenous base, where is its position, where exactly is its coding for what function in our body, that entire study has to be done. So the branch of biology that deals with the study of genes is called genetics. So it, what does it help us? It helps us to study the genes in detail. So our chapter is basically on how we will be studying the genes. So this is the importance why the chapter of genetics is included in your curriculum, so as to know that how the characteristics are passed from generation to generation. Now, who was the person who studied about genes? His name was John Gregor Mendel. So he's known as the father of genetics, and he was the person who understood that it is through something. It is the DNA. It is the DNA is having something inside, and this is why what we know that Watson and Crick confirmed that it is something in the DNA which helps us to pass from one generation to the other. On basis of Watson and Crick's theory, John Gregor Mendel proposed some more experiments and concluded, yes, in fact and indeed, there are some traits and the genes are responsible and the genes are present inside the DNA. They went on more deeper level. They, from the cell, the nucleus to chromosome. Inside the chromosome, we are having DNA. Inside the DNA, we are having genes. So now this Gregor Mendel started studying inside the gene and what is happening when two genes or maybe three genes are combining with each other, and how they are mixing, and whether we can form a new character or something like that. So what did he do? He studied these experiments on a pea plant. So what was this? The first uh, like plant or first model of study, it was a pea plant. And why he selected a pea plant? There are many organisms. He could have started working on animals, or he would have started even on some a different plant. He started his studies on a pea plant. Why a pea plant? Because a pea plant is very easy to grow. Second, at that time when he started his studies, they are self-pollinating, but also can be cross-pollinated. That means a pea plant is such a organism of study that it can also do self pollination. That means the gametes are present on the same plant, or if he wants to mix the characteristics, he can also take another species of a pea plant, because there are several characteristics in a pea plant. He could also study by doing cross pollination. It is an annual plant. That means it grows all round the year, and many generations can be studied within a short period of time. So, supposing if there is a plant which only grows in a summer season, so I have to wait till the next summer season to study more about this plant. Since this is an annual plant, I can perform several experiments and I can keep growing the plants in the number I want, and I can check, I can mix match, I can do self pollination and do whatever I am getting with my self pollination. I can always do cross pollination, right? So this was the reason why he selected pea plant. And the most important was that in the pea plant itself, there were many contrasting characteristics. Like it was, he found one pea plant to be tall, one pea plant to be short, one to be green, one to be yellow, and so forth. Now, what do the experiments Mendel do? Mendel experimented because he wanted to prove that genes are responsible for characteristics to pass from one generation to the other. So he proved that there is some factor which is getting inherited, right? So he performed his experiment in two ways. First one was called a monohybrid cross, and a second one was called a dihybrid cross, right? So while experimenting, he found that certain factors has he already knew. So he performed, and while he performed the experiment, he saw that yes, certain factors definitely passed from offspring in a confirmed and in a stable way. Yes, it was getting transferred. Now, these factors, whatever he found in the offspring, are nothing called genes, and he termed genes as the unit of inheritance. Now, let's come to what is a monohybrid cross. So, what he did Mendel do? He took a tall pea plant and he took a dwarf pea plant, right? And he did a crossing over. You don't know what is a crossing over? Yes, two homologous chromosomes. He did a crossing over. Now, when he did a crossing over, he found that the gametes from the tall plant, that is, it is represented by capital T. We usually have some more importance why we use this capital T and a small t. It is because whenever we are putting a capital T, it is something like it is a dominant characteristic. That means it is going to confirm, it is going to, while crossing over, it is going to show its dominance, that it is going to show its power. That means I am dominant, right? And the other small t, it is called something called recessive. What is it called? Recessive. That is, while the while the generation after generation is continuing, the recessive part of the gene will be slightly less powerful than the dominant one. We will be learning more and more on a later phase. But let's see what did he do in this monohybrid cross. Monohybrid cross, he took a tall plant and he took a dwarf plant. And the first, after this is the parent generation. P is called the parent generation. So in the first gamete cross, which is called the F1 progeny, that is the what did he find in the first cross? He got something called T and T. Since it's a mixed match of a tall and a dwarf, they say it is a hybrid version. It's a hybrid version. So it is a monohybrid offspring. It is having a tall also and a dwarf also. Now, what did Mendel do after this? He took these T and T and just tried to separate the gametes. Okay, and this he made a box. This box is called a Punnett square. It is called a Punnett square. Now he tried to self cross them. That means the T, the gametes T and T and T and T are selfing with each other. When he selfed them, he found that this, see, this is the way T, T, T and T, you just multiply in this way. So with this, whenever there is a capital T, it means that the offspring will always be tall. So this, the offspring in the first box is the tall one. Here in this second box also we are having a capital C. This offspring will also be a tall one. See, this one is also a tall one, but we see two similar organisms this way. Yes, and we are finding a fourth box where we see this small dwarf and another gamete of a dwarf connects, and we are getting a dwarf organism. So we are getting a homozygous tall. That means both the gametes are same. So we will come across a word called allele in our later classes. So we'll come to know. So these two homologous tall is one. But we are getting a mixed match, which we call in a biological way heterozygous. That means the chromosomes are different. So they are two in number. And then we get a fourth one, which is also homozygous, but it's a dwarf. So we get our offspring in the ratio of 3:1. So what did a monohybrid cross yield? Monohybrid cross yield 3 tall plants and 1 dwarf plant. So let's see. See, this is what I explained to you. He found that the first offsprings were tall and called it F progeny. Then he crossed the F progeny and obtained both tall and short plants. Right? So this is how if we keep on crossing, we can get tall and dwarf plants also. So if he found this, he also did this monohybrid cross with other characteristics of the pea plant. So what are they? Mendel even conducted experiment with green peas versus yellow peas, round versus wrinkled, and etc. Yeah? So are we clear what is a monohybrid cross? Yes. So now we come. So in all the cases when he did this, all these other contrasting traits, all the results were same. Three of the, like suppose it's a green pea versus yellow pea, you would find green, if the green pea is having a dominant allele, we will find three green pea plants and one yellow pea. Clear? Now the second one was a dihybrid cross. Now let's see what is a dihybrid cross. Mendel consisted two traits. Yeah, suppose like green and wrinkled. There it was just tall and dwarf. Here he mixed two characteristics at one time and he crossed two characteristics with two characteristics. Now let's see what did he do. He crossed green wrinkled seeds with round and yellow seeds. That means he, what we can find it very clear from these words is the shape of the seed and the color of the seed. So we are having two characteristics, shape and color. So what did he find? He observed that all the first generation progeny was round and yellow. Let's see. So what did he do? He took a green wrinkle and he took a round yellow, right? And what did he do? He did this Punnett square and he found that he found all of them to be having a round yellow. This meant that the dominant traits were the round yellow and round shaped and yellow colored. So what do we find from here? We find that since he found all of them to be yellow and round, this is the dominant version. Now, what did he do with the F1 progeny? He again crossed them. See, all these F1 progeny, these are the he again self-crossed them. And when he crossed them, he found a combination of all the characteristics. So what are the combinations he found? He found wrinkled yellow, he found round yellow, he found wrinkled green, and he also found round green. And all of them, see, are in the ratio of 9. See, 9, how much we are finding? We are finding round yellow, how many? Wrinkled yellow, see. 3:1. Yeah. So what are the conclusions Mendel got from this? He found three laws from these two experiments, that is the monohybrid cross and the dihybrid cross. He found the law of dominance, law of segregation, and law of independent assortment. So these we will be studying in a detail in our next class, and I hope you enjoyed my session on a brief intro on what exactly is genetics and how we actually come across how the genes are transferred from one generation to the other. Thank you.